Inside every single one of the 37 trillion cells in your body right now, a microscopic power struggle is constantly taking place. At the very center of this complex metabolic process is a critical coenzyme called nicotinamide adenine dinucleotide, commonly abbreviated as NAD+. Without this single essential coenzyme, cellular respiration stops completely, cellular repair mechanisms fail instantly, and human life ceases within seconds. Yet, modern scientific research reveals a troubling biological reality. As we age, our natural systemic levels of NAD+ decline precipitously across virtually every tissue. By the time the average adult reaches 40 years of age, their systemic NAD levels have dropped by nearly 50% compared to early youth. By age 60, cellular concentrations of NAD+ can fall to less than 20% of their peak youthful baseline. This profound biological drop raises a pivotal scientific question. Is the decline of NAD+ merely a passive symptom of aging, or is it the primary engine driving cellular breakdown? To understand why this single coenzyme matters so much for longevity, we must first look inside the mitochondria, the internal cellular power plants responsible for generating energy. Mitochondria convert the nutrient molecules from the food you consume into adenosine triphosphate, the universal bioenergetic currency known as ATP. However, the fundamental chemical reactions of the electron transport chain inside mitochondria cannot function without NAD+ constantly accepting and donating high-energy electrons. When NAD levels drop inside the mitochondria, cellular energy production slows down drastically, leaving tissues fatigued, impaired, and highly vulnerable to oxidative stress. Beyond simple bioenergetic energy generation, NAD+ acts as a vital signaling fuel for a crucial family of regulatory enzymes known as sirtuins. Sirtuins are specialized deacetylase proteins that regulate chromatin structure, suppress cellular inflammation, promote mitochondrial biogenesis, and repair damaged DNA strands throughout the genome. Biologists often refer to sirtuins as the primary guardians of the cell, but these protective enzymes are strictly and non-negotiably NAD dependent. If cellular NAD+ levels drop too low, sirtuin enzymes remain completely dormant and inactive, leaving your genomic DNA unprotected against cumulative molecular damage. A second major metabolic consumer of NAD+ inside the cell is a family of genomic repair enzymes known as poly-ADP ribose polymerases, or PARPs. Whenever solar radiation, environmental toxins, or normal oxidative metabolic stress causes single-strand breaks in your DNA, PRP enzymes consume vast quantities of NAD to repair the damage. As we age and accumulate more chronic cellular stress, overactivated PRP enzymes can rapidly drain the cell's remaining NAD pool, competing directly with sirtuins for survival resources. This chronic biochemical drain creates a vicious biological cycle where lower NAD leads to poorer DNA repair, which accelerates cellular aging and further depletes NAD supplies. This critical discovery led modern longevity researchers to ask whether restoring systemic NAD+ levels could reactivate cellular repair mechanisms and support metabolic health. However, directly consuming or injecting raw NAD+ presents significant physiological challenges because the molecule is far too large and unstable to cross intact cell membranes easily. Instead, modern biochemical research has focused heavily on cellular precursors, smaller building block molecules that cells can readily absorb and convert into functional NAD+ internally. The most heavily studied precursor in human clinical trials today is nicotinamide mononucleotide, commonly abbreviated as NMN. NMN is a naturally occurring nucleotide derivative formed from vitamin B3 and phosphate within human metabolic pathways. When ingested orally, NMN enters bloodstream circulation and is transported directly into cells through dedicated cell membrane transporters like SLC12A8. Once inside the cellular cytoplasm, enzymes rapidly convert NMN into active NAD+ through a single enzymatic step catalyzed by NMN adenyltransferases. Human clinical trials conducted over the past several years have begun evaluating how oral NMN supplementation affects human physiological markers under controlled conditions. A landmark 10-week clinical trial conducted at Washington University School of Medicine examined the metabolic effects of NMN in postmenopausal women with prediabetes. The clinical researchers observed that 500 milligrams of daily NMN supplementation significantly improved muscle insulin sensitivity and enhanced cellular glucose uptake capacity. Another randomized, double-blind clinical trial published in 2021 evaluated physical endurance and aerobic capacity in middle-aged runners taking daily NMN supplements. The scientific findings demonstrated that combining regular exercise with NMN supplementation increased oxygen utilization efficiency and cardiovascular performance significantly more than exercise alone. Further human trials conducted in Japan and the United States have consistently shown that daily oral NMN doses between 250 and 1,200 milligrams reliably elevate blood plasma NAD levels. These systemic increases in NAD+ are accompanied by measurable improvements in vascular endothelial function and arterial flexibility in older adults. Vascular aging is characterized by the progressive loss of nitric oxide production and stiffening of blood vessel walls, which limits oxygen and nutrient delivery to vital organs. By restoring endothelial NAD levels, sirtuin 1 activity increases, promoting new capillary growth and restoring youthful blood flow to ischemic tissues throughout the body. In addition to cardiovascular benefits, emerging neurobiological research suggests that maintaining mitochondrial NAD levels may protect delicate brain neurons against neurodegenerative decline. Preclinical studies indicate that NMN supplementation helps preserve synaptic density, reduces neuroinflammation, and supports cognitive processing under conditions of metabolic stress. Furthermore, researchers have uncovered how NAD+ influences circadian rhythms and sleep-wake cycles through interaction with core clock proteins like clock and BMAL1. Sirtuin 1 regulates the molecular clock in the hypothalamus, meaning that declining NAD levels can disrupt deep sleep architecture and hormone regulation as we age. Restoring NAD through precursors helps realign circadian gene expression, leading to improved nocturnal sleep quality and daytime metabolic alertness. However, scientific rigor requires looking closely at alternative NAD precursors and understanding the subtle biochemical differences between them. Nicotinamide riboside, or NR, is another widely studied precursor that follows a closely related biochemical pathway to NMN. NR must first be phosphorylated inside the cell by nicotinamide riboside kinase enzymes to become NMN before it can finally transform into active NAD+. Both precursors effectively raise cellular NAD levels in clinical testing, but bioenergetic researchers continue to debate which molecule offers superior tissue-specific absorption. Another traditional precursor is basic niacin, or vitamin B3, which elevates NAD through the Price-Handler pathway, but often triggers uncomfortable skin flushing at therapeutic doses. Dietary sources also contain trace amounts of NMN, including broccoli, cabbage, cucumbers, edamame, and avocados, though consuming therapeutic amounts through food alone is practically impossible. While the clinical evidence surrounding NMN is highly promising, responsible health science demands a balanced evaluation of potential risks, quality controls, and dosage considerations. Long-term human safety studies extending beyond 1 year are still ongoing, meaning the long-term cellular effects of sustained high-dose precursor supplementation are not yet fully charted. Furthermore, because NAD+ powers rapid cellular division, some researchers caution that excessive NAD elevation could theoretically fuel the metabolic demands of existing subclinical tumors. Supplement quality presents another major challenge in the commercial wellness market, as independent third-party lab testing frequently reveals mislabeled or underdosed products. Consumers interested in precursor science should prioritize verified liposomal or pharmaceutical-grade formulations tested for purity, heavy metals, microplastics, and active ingredient stability. Beyond oral supplementation, lifestyle interventions remain powerful, scientifically proven methods to naturally preserve and boost your body's NAD pool. Regular aerobic exercise and high-intensity interval training stimulate the enzyme NAMPT, which recycles degraded nicotinamide back into fresh NMN naturally. Intermittent fasting and caloric restriction also activate cellular NAD synthesis pathways by mimicking nutrient scarcity and triggering sirtuin activation. Optimizing sleep hygiene and minimizing chronic alcohol consumption protect NAD pools by preventing systemic inflammatory overload and liver enzyme depletion. Stress management techniques like meditation and breath work lower stress hormone cortisol, reducing unnecessary PRP enzyme activation and preserving cellular NAD stores. Ultimately, NMN and NAD biology represent one of the most exciting frontiers in contemporary cellular health and longevity science. As clinical research continues to expand, understanding how to support your cellular power plants becomes an essential component of evidence-based health optimization. Please remember that this video provides general scientific information for educational purposes and is not medical advice, so always consult a qualified doctor regarding your personal health. If you found this scientific deep dive insightful, please like the video, subscribe to